previous slides were not clear enough. I hope that this slide clarifies this two-hit model. So this is retinoblastoma, which is a cancer that affects retina in the eye, and it's the most common form of cancer, like ocular cancer among children. And it may occur in this Mendelian or familial case, but it may also occur as sporadic. So now I think this slide explains the difference well. So let's start with the pedigree. So here we see a pedigree that even without knowing what the condition is, you would say that this is likely autosomal dominant effect, right? Because it occurs in every generation and affects both sexes. Affected individuals have at least one affected parent. And indeed, this is something that we observe in the case of familial retinoblastoma. This is in comparison with sporadic cancer. So we see that one girl here got retinoblastoma, but there is no history of this cancer in the family, which makes it a sporadic case. So this is sporadic, contrasted with Mendelian or familial case. So what's going on here? What's the difference? Why one family has this increase of this condition, but in sporadic cases we don't observe it? So it's because of this germline mutation that the first mutation is inherited from parents, like, for example, this person, this female, might be a carrier, and she may never develop the cancer, but the risk is higher because it takes only a second mutation in somatic cells in the eye to result in retinoblastoma. And because it doesn't take much to acquire the second mutation, we see not only increased frequency of the disease in the family, but also the presentation is more severe. So we see like multiple tumors. It could be bilateral, which means that not just one eye, because like in sporadic cases, it's only one eye affected, but it could be both eyes affected. Or there could be earlier onset of this disease than normal, also suggesting that the severity is increased, which is caused by this Mendelian case, this pattern of inheritance. So again, this is in contrast with sporadic, that requires two somatic mutations, which are less so. So you may expect one, which is very rare, but then the second is super rare. That's why the sporadic cancer is sporadic by definition, right? It's also retinoblastoma, and this is something. So first it was suggested as a model, and it was actually the first model was based on retinoblastoma, but then it was proven because indeed we in the affected tissue we see loss of heterozygosity, which basically we see both alleles mutated. Various mechanisms leading to the loss of function can be epigenetic silencing, mutation, somatic recombination, loss and duplication, all chromosomal loss, that results in the second hit. We can visualize the chromosome. So this is chromosome 13, and the region where the gene is located is 13q14. So this is an example of a patient with retinoblastoma that has a deletion in this right-hand homolog, leading to the cancer. Now, so that's retinoblastoma, but it applies to other tumor suppressor genes, and I'll show you more syndromes in just a second. There's more syndromes like that. But now going back to the table, I'd like to, where's the table? Oh, it's actually in the previous. The table with tumor suppressor genes. Or before we go to the table, BRCA1 and BRCA2, you may have noticed that they classified as autosomal dominant transmission, right? And when you look at the familial cases, this is indeed the case. Like this retinoblastoma case, we had in the familial case, in the Mendelian case, we had a pattern that was consistent with autosomal dominance, right? And why is that? Why are those tumor suppressor genes listed as autosomal dominant conditions, even though it takes 2 mutations for loss of function in the locus? It's because when we test a patient with for being a BRCA1 or BRCA2 carrier, we take random samples of, typically it's blood sample, right? So we look at germline genotype. So we take a blood sample and genotype it, and we see that the person is either a carrier or is not, right? Now, there's, from the table that I showed you before, this one, it's clear there's like 50% increase in risk for a person, risk of cancer for a person being a carrier. So even though she's a carrier, she may or may not develop the cancer. So you could see, so there are two concepts that apply here. So one is autosomal dominant inheritance based on the genotype that we see based on a blood sample, and reduced penetrance, because if a person doesn't develop, if a carrier doesn't develop breast cancer, we say that this is reduced penetrance, right? But the tricky part here is the mechanism itself, because when you look at the mechanism, again, I'm using retinoblastoma as an example. The mechanism itself requires total loss of function. It requires, it tells us that this is about, the effect itself is recessive. Okay, so I know it's confusing because it's not a true dominance. We classify the autosomal dominance based on the germline genotype, but the true genotype within the cancer is recessive homozygote. So it is recessive with tumor suppressor genes. When I said that oncogenes have dominant effects, the mutations have dominant effects or gain of function, and tumor suppressor genes have recessive effects, it still applies with this exception, with this confusing classification of breast cancer and other familial cases like that as autosomal dominant conditions. So if you have a question, what is the inheritance mode or inheritance pattern of breast cancer with when this BRCA1 or BRCA2 genes are involved? The answer is autosomal dominant, because this is the standard classification of these diseases. Does that make sense? Even though the true mechanism is actually loss of function in the cancer itself. So that's a very important distinction and case that I wanted to make. So here's another hereditary cancer, and because it affects some families, and the presentation is quite severe. It acquired its own name. It's von Hippel-Lindau disease or syndrome, and we know what the gene, the major gene involved is. So this is VHL. And what's characteristic with this von Hippel-Lindau disease is that, again, when we look at that, this is autosomal dominant disorder, but the presentation in various members of the family are different. So they all get cancer, but different types of cancer. So one of them has retinal hemangioblastoma, another has cerebellar hemangioblastoma, kidney cancer, and pancreatic cancer. Sometimes the same person may have different, two different types of cancer. So this is due to this pathway controlled by VHL, which is vascular endothelial growth factor. Here's another. Oh yeah, I forgot about the question. I'm sorry.
What's the last slide? Was that due to an error that put off the gene?
The last slide.
Was that with GHL? Or is that still the mutation that causes it?
Right. Yes. Oncogenes?
Yeah.
No, because that's a good question. Because I mean, of course, you can imagine a situation like that, right? That oncogene mutation is inherited from the germline, but because these effects are so severe. That when they show up in embryogenesis, these effects are lethal. Because think about it, when a mutation in oncogene happens, it happens typically late in life and it leads to cancer. Now imagine what would happen if it occurs very early in development. So it's more by assumption, but this is what happens, that that mutations in oncogenes are not inherited because they're so deleterious that they kill the embryo. This is Lee-Fraumeni syndrome family, which is due to complete deletion of p53. So I showed you how important the p53 gene is. It's the guardian of the genome. It responds to tumors and cancers as long as it's not mutated itself. But when it's missing, when it's again, this is p53 is the tumor suppressor gene. So if one of the copies is mutated in the germline, then it's relatively easy to acquire the second somatic hit, the somatic mutation, which result in these various types of cancers, like breast cancer, leukemias, and brain tumor, affecting a variety of tissues, not just one tissue, but tissues across the board. And this is Lynch syndrome, another hereditary, yes.
One question from the previous slide. So for the second generation, they did the circle. So basically we have the hereditary and then it happens sporadic at the same time. That's why he's
You mean yellow?
no, the affected was cancer.
This one?
The second one, the 41. Yeah, this one. So he basically inherited from the parent and the sporadic happened at the
Yes,
same time.
yes.
That was my question.
So in a sense, I mean, we don't call them carriers, even though they have the germline mutation. That we don't call them carriers because they are affected, right? This person is a carrier, like a true carrier, because it has that germline mutation, but it's not affected. So in this case, we would, in a sense, this is reduced penetrance in this particular case, right?
Yeah, my question is for this one, the one who was affected.
Like this female.
Yeah, was it, it's just him getting it by the, like,
The second hit is somatic. The first mutation is germline. And this is Lynch syndrome, which is caused by defects of DNA repair genes like MSH1, MSH2, and H6, TMS2, and ITCAN. These genes, or this defect is related to colon cancer, and it's called hereditary non-polyposis colon cancer, HNPCC. Here, there's significant risk increase, so it's 80% risk in lifetime. But in a sense, we're talking about inheritance of the risk rather than the cancer, because there's 20% chance that the person won't develop a cancer even though he or she is a carrier. In which case, again, it would be reduced penetrance. So there are some families in which there is inherited cancer susceptibility, and we can tell that this is the case, not sporadic, but based on these characteristics. So for example, if several close first or second degree relatives have a common cancer, again, this is suggestive of familial case. It also applies to cases when several close relatives with different have different related cancers, like breast and ovary cancers, or when two family members have the same rare type of cancer. Because it's so rare in the population, when relatives have it, again, it suggests that there's something going on in the family that it's inherited. And then there's some criteria that apply to the symptoms, the presentation. So for example, there could be unusual early age of onset. So I mentioned that in the context of retinoblastoma, that a sporadic case affects children, but if it's like super early, unusually early case, it could be suggestive of familial case. Or bilateral tumors in paired organs, like, for example, breast cancer, not in one breast, but both breasts, or retinoblastoma, not in one eye, but both eyes. Synchronous or successive tumors, or tumors in two different organ systems in one individual. This is also very unusual, suggesting that there is more than just somatic mutations. That there were some mutations, some susceptibility mutations inherited in the germline. And that leads me to metastasis, which is the most scary phase of cancer, because this is what cancer patients die of in 90%. So the curse of death in cancer in 90% cases is metastasis itself, which is, so this is a process in which epithelial cells lose their cell polarity and cell-cell adhesion. They detach from the primary tumor and gain this, they acquire these migratory and invasive properties to become mesenchymal stem cells. So there's some reprogramming involved, that they are kind of reprogrammed back to their stem cell, non-differentiated cellular state. And this is known as EMT, so I want you to be aware of it. This EMT process, this epithelial mesenchymal transition is very important in metastasis. So these cells, these migratory cells that undergo EMT, become multipotent, undifferentiated cells that can now invade many types of tissues and differentiate there and grow into independent metastatic lesions. This slide shows you common metastatic distribution. So it turns out that some cancers are more invasive than others in terms of metastasis. So for example, breast cancer is super invasive, but most commonly the secondary lesions appear in liver, in brain, or bones, or they also metastasize to lung. This diagram shows you actually metastatic sites and their primary origin. So breast cancer is in yellow, and you can see that actually the whole diagram is dominated by yellow color. So it shows you how aggressive the breast cancer is. It invades many other organs. So, for example, even for lung cancer, so this is lung cancer. So when we look at the cells that end up in lung, so this is identified or diagnosed as lung cancer. But when we look at exactly where the cells are. In many cases, they actually come from the primary tumor in breast. Ovarian cancer metastasizes into pleura or liver. Colorectal cancer typically invades liver. What else do we have here? Lung cancer goes to bone or brain. Skin melanoma may invade brain as well. So there are some patterns there, but like I said, some primary tumors are more aggressive than others. This is a very important process in cancer development known as tumor angiogenesis, which is growth of vascular vessels within tumors. Turns out that when tumor grows beyond 1 or 2 millimeters in diameter, it requires independent supply of blood. So it needs to recruit blood vessels to grow further. Otherwise, there is no oxygen, no nutrients, and such a tumor will either shrink or just stay as is, but it won't grow further. So this is this process of angiogenesis is driven by this VEGF, vascular endothelial growth factor, and it's encoded by a gene VEGF gene that stimulates this vascular growth. So it's a necessary part of the process in the progression of cancer from small localized neoplasms to larger growing and potentially metastatic tumors. So this is tumor, and this is you can see the red structure here is the blood vessel. So the tumor recruits these blood vessels. But here's another striking thing, which is a discovery of like the last 2 or 3 years, and it's really mind-blowing. Turns out that there is some crosstalk, there's some communication, not just between tumors and blood vessels, but between tumors and nerves. So it was noticed that when a nerve cell was put in a petri dish next to cancerous cells, they started to reach out to each other. So the nerve cells were reaching out to the tumor, and the tumor was stimulated, and the cells were growing, they were dividing further. So there was some, like I said, crosstalk promoting the growth and spread of cancer. And this is something that we just started to not just understand, but started to notice. So we know that nerves transmit these signals through norepinephrine, and this turns out that the signal norepinephrine is stimulating further growth of cancer. It also stimulates receptors and endothelial cells that make up blood vessels. So there is an extra recruitment of blood vessels due to this interaction between tumor and nerve cells. Then there's some, turns out that nerves also provide like immune protection because they send a signal to immune cells to stand down. That's another amazing observation. And finally, and maybe this is the most striking thing, that cancer cells themselves can produce neurotrophic factors, molecules that encourage nerves to branch out and elongate toward the tumor. So it's not just, so they kind of hijack, they kind of steal the mechanism from nerves to stimulate its own growth. So this could be a link between stress and cancer that was kind of suspected but ignored by modern medicine because we didn't really have a mechanism, we didn't really have evidence for this crosstalk, but now the picture is emerging, and here is a person, cancer researcher, Elizabeth Ripaski, from Roswell Park Comprehensive Cancer Center in Buffalo. She says, the idea that tumors can be so controlled by these nerves, all of a sudden, it really brings some clarity into why various types of stress can be so bad for people. Okay, so that concludes cancer genetics.